Question number 15. Now we are given a diagram and asked to answer question 15 and question 16. YAMB, question 15 now. YAMB is used in the setup because it what? YAMB is used in the setup because it acts as a semi-permeable membrane. It acts as a semi-permeable membrane. A semi-permeable membrane allows small particles to pass through it, but the larger ones, it doesn't allow it. So, YAMB is used in the setup because it is made up of cells with membrane, which selectively allow passage of small molecules like water, disallowing large misrule like soil particles. So you see, it allows small particles and then it holds back the large particles. So YAMB in the setup is used mainly, which acts mainly as a semi-permeable membrane. Question number 16. Which of the following results is to be expected if the setup is left for several hours? If the setup is left for several hours, the setup that is likely, the expected setup will now be decrease in the volume of water. Decrease in the volume of water inside the YAMB. Now, if the setup is left for several hours, there will be a decrease in the volume of water in the YAMB cavity as a rule, as a result of osmosis. Note that as a result of what? Osmosis. So the salt solution in the beaker is of higher osmotic concentration than water, and they exert higher osmotic pressure that draws water molecule from the YAMB cavity across the cell molecule from the YAMB cavity across the cell membrane that is semi-permeable. That is what? Semi-permeable. So if the setup is allowed for hours, it decreases the volume of water inside the YAMB. Question number 17. The surface of an alveolus in a mammal is well supplied with tiny blood vessels, known as what? Known as the capillaries. Capillaries are the first blood vessel which branch from the arteries. Now, several arterioles units to form several arterioles, which are the small arteries, unite to form the arteries. Now, capillaries later unite to form the venules, which unite to form the veins. That sends deoxygenated blood back to the heart. So they move in pari passu in order to enhance blood flow in the body of the organisms. Question number 18. The anaerobic fermentation of a glucose molecule yield what? The anaerobic fermentation of glucose molecule yield pyruvic acid and alcohol. Now, aerobic fermentation of glucose is a type of fermentation in which oxygen is used to oxidize a sugar molecule inside the cell with the release of energy, carbon dioxide, and water with 38 ATP. Now you see, while anaerobic fermentation is a type of fermentation which involves complete absence of what? Oxygen to yield 2 ATP and ethanol. 2 ATP and what? Ethanol. So anaerobic fermentation of glucose molecule result or yield to what? Pyruvic acid and alcohol. Question number 19. The opening of the stoma is controlled by the dash. The opening of the stoma is controlled by the increase in solute concentration in the guard cell. The increase in solute concentration in the guard cell. Now, guard cell is a cell found in the lower epidermis of the plant that determines the opening and the closing of the stomata. Now, increasing in solute concentration in the guard cell leads to opening of the stomata, while increase in the solute concentration leads to closing of the stomata. So the solute concentration of the guard cell determines either the stomata will open or close. So when the concentration is increased, the stomata tends to open. When the concentration is lowered, the stomata tends to what? Be low. So the answer to the question is increase in solute concentration in the guard cell. Question number 20. In question number 20, we are given a diagram. And this diagram, we are instructed to answer question 20 and 21. The sweat gland, that's number 20 now. The sweat gland is a structure labeled dash. The sweat gland is a structure labeled IV. That is a sweat gland. The sweat gland is a structure labeled what? IV. Now, sweat gland is an opening through the skin in which sweat reaches the skin surface from sweat gland. It is located in the part labeled IV.